Welcome my dear students. Today's topic is Progymnosperm and evolution of Megasporangia. Look at your screen. In the screen there is a Megasporangia with many megaspores. This figure is represented by Archaeopteryx. Okay. Now let us begin. Cloner and Petit in 1987 have explained how do the Progymnosperm play an important role in evolution of Megasporangium, and they represent a series of events. In their series, the first stage is represented by Archaeopteryx. Again, I am quoting, the first figure represents the sporangium of Archaeopteryx with many megaspores. Now we are moving to the second figure. Look at the figure. It is the Megasporangium of Bothrodendrostrobus. In this figure there are only 4 megaspores and these are of equal sizes, and all these 4 are functional. So we may say that the first step of evolution is the reduction of the number of megaspores. In Archaeopteryx there were many megaspores inside a Megasporangium, but due to evolution the number reduces in Bothrodendrostrobus and it is only 4 in number, and all these 4 are equal in size and all are functional. Now we are moving to the figure number 3. This figure is a representation of the Megasporangium of Bansonitis. Here the further course of evolution is observed in the Megasporangium, where the number of megaspores are as same as the second case. But it is to be noted that in this case 2 megaspores are large in size. In your screen it is yellow in color in the figure number 3, and the remaining 2 are reduced and they are smaller in comparison to the first 2. Okay. Now we are representing, or we are going to discuss about the figure number 4, that is the Megasporangium of Archaeosperma arnoldi. Look at the first figure. Here the number of megaspores are many. That is, Archaeopteryx represents many megaspores in the Megasporangium, but the number reduced in 4 in case of Bothrodendrostrobus. And interestingly, in the third figure, that is in Bansonitis, 2 are larger and 2 are smaller. Now look at the figure number 4. Here the number of megaspores are 4, but the sizes are of different. Okay, the top 3 are smaller and a large megaspore is observed in the central position, okay, occupying the whole embryo sac. So we may say the next step of evolution is shown by Archaeosperma, where the number of megaspores are 4, but 3 are greatly reduced in their size, and perhaps these 3 are the non-functional, and the megaspores are tetrahedral. The ovule had a spiny integument. Look at the integument. Few spines are there, which, that is, the integument at the distal end forms a micropyle. Now, what is about the nucellus? Due to poor preservation during the fossilization process, the nucellar tissue could not be studied in Archaeosperma arnoldi. Hence, we cannot say anything about the nucellus of this ovule. That is, in the ovule of Archaeosperma, the nucellus is lack, and it is only due to the poor preservation process. Now, we have to study the figure number 5. In this case, more evolved Megasporangia is observed. It is the ovule of Cardiocarpus. Here the number of megaspores are 4, same as that of Bensonetis, Archaeosperma in the number of megaspores. But 3 of them at the distal micropylar end are much more smaller than those of Archaeosperma. Please compare the figure number 4 and figure number 5. The number of megaspores are same. But in case of Archaeosperma, the top 3 are much more larger than the top 3 of Cardiocarpus. Here also, these 3 small megaspores are non-functional. Now, look at your screen. There are a series of ovules or megasporangiums. The first one is from Archaeopteris, second one is from Bothrodiendrostrobus, third one is from Bensonetis, fourth one is from Archaeosperma, fifth one is from Cardiocarpus, and the sixth one, the last one, that is at the corner of the right hand side, top one, that is the ovule of Cycas. Here, in the last stage of this evolutionary series, the ovule of Cycas represents only one functional megaspore, and it is of much enlarged size. And what about the other 3? The other 3 ephemeral non-functional megaspores quickly degenerate. And here the megaspores, before their degeneration, they were arranged in a line, that is, they arranged as linear tetrad. Here look at the figure. Here the top 3 are degenerating, hence they are represented by just 3 lines, and the functional one is the large one, and it will take part in the formation of the female gametophyte. So by this morphological series, the scientists, they represent different stages in the reduction of megaspore number to a single tetrad, that is, from numerous number of Archaeopteris to a single one in case of Cycas. Okay, that is in the Paleozoic plants, and again I am saying the series was started with Archaeopteris and it ends to extant genus Cycas. So again study the figures carefully. 6 figures are there. In the first figure there are numerous megaspores. In the second one, the number has been reduced. From second to the sixth, the number are 4. But in the last one, the only one is the functional and rest 3 degenerated. Okay. So this is about the role of progymnosperm in evolution of Megasporangium. Hope again this will help you to study the paleobotany, that is the fossil plants. Thank you.